This is Me Before Mom, the podcast that helps moms stay connected to themselves while doing the most important job they'll ever have. And I'm your host, Bert Anderson. Hey, it's Bert, and you are listening to Me Before Mom. Today, we have a fun guest. Well, fun for me. I don't know if it's fun for you, but we're going to talk to my brilliant brother-in-law, my little sister's husband, Dr. James A. Ankrum. You sound so professional when I say that.
I am professional.
I know you've been to enough school. You've done enough things, and you've built a human heart. I'm just kidding. You haven't. I learned my lesson last time. So James is a biomedical engineer. He works at the University of Minnesota. Just kidding. Iowa, guys. Sorry.
I did work at University of Minnesota.
You did.
For a year.
And I was thinking about all of your schooling.
Yeah. For the last 6, I've been at University of Iowa.
Yeah. I won't make that mistake again. I know. Hawkeyes, Gophers.
One's better. We all know which one.
I know. The one who won last year. Thank you very much.
Which was Iowa.
That's right. I forgot about that. You pay way more attention to college football than I do. I don't care about that. But anyways, guys, so during this pandemic, COVID, all that fun stuff, I have found myself frequently calling James, either freaking out about something I've read on the internet or some video I've seen on Facebook and being like, oh my gosh, I don't know what to do. Like, what do I believe? So James has become the voice of reason in my head, which is so fun. So James, can you give us a little bit of background on what you do and how you got here?
Sure. So I'm an associate professor of biomedical engineering, which means I teach classes, both undergraduate and graduate courses in biomedical engineering, including a class on scientific communication, which involves a lot of how do you process complex literature from medicine and science. And I also run a research lab within the Fraternal Order of Eagles Diabetes Research Center at the University of Iowa. So we do work on cell-based therapeutics, which are kind of a fancy next generation therapy, as well as developing organoids, which are little micro tissues that we grow in the lab to screen drugs and toxins for their effects on human health.
Oh, interesting.
So I got here through way too much school. So
much school.
So much school.
James is from a really tiny little town in Iowa.
Yes.
Eldora.
Yep.
I used to say that he's like the farm boy who went and became this big fancy PhD person, but then he reminds me he's not an Iowa farm boy.
I grew up across the street from a farm, but not on a farm.
Well, that kind of to me is a little Iowa farm boyish, okay?
All Iowa small towns are surrounded by farms.
That is true. And cornfields. Lots and lots of cornfields.
Yes. And beans.
Beans, that's right. Can't forget about the beans. So, James, let's talk about how do we actually read the scientific communication that's coming out?
Yeah, so I think one thing that's different probably from the way that scientists go about reading about science and medicine news in the lay public is scientists seldom, if ever, use any kind of highly processed media stream. But what I mean by that is, your nightly news for headlights, like that might be kind of an appetizer, something piques our interest, but we never leave it at that. We always want to dig deeper into it. And so typically anything you hear on the news, there is a full study behind it. There's a paper somewhere, there's something on the internet that was the original source material for whatever's being claimed in the news. And so getting in the habit, when you do hear something that's confusing or hard to believe, or you want to know, is it really that promising? That source material is typically something you can track down. So that's one thing that we do. So I find that that's really helpful, because sometimes you'll hear on the news, getting away from COVID, but just the things we've all heard of, like, 6 cups of coffee, improves longevity. You
And
hear
if you
this
guys
on,
wanted to know, James is holding up his
yeah,
cup of coffee.
and you hear that and you're like, wow, like I feel so validated in my coffee habit. But then if you go and read the study, you find that it was actually, a study done with 12 rats, and it, like, it was like there was all this interpretation that happened from the initial study to the actual news blurb. And so that's not as strong of evidence, right, to say that I should suddenly up my caffeine intake by a few cups a day.
Yeah.
Whereas there's others that you hear it and you go and look at the actual original study and you realize, oh my goodness, this was a study of 10,000 patients, really rigorous, like this is really high quality, like, data. The results are really
Right.
strong. And so being able to ask, well, where did this claim come from, I think is a good follow-up question anytime you hear a claim on the news. Then either try to look for some of the original source material or even some of the slightly less processed. So I've talked to you about this, but my go-to science news site, I got a couple of them, but Stat News is one that I really like. All their coronavirus stuff is free right now.
Mm-hmm.
They still process it for you so you can understand it as a layperson, but they don't strip away all the qualifications of,
Right.
it was a limited size study, it's only in phase 1, all these milestones still need to be done. So the level of analysis is a lot deeper than just potential breakthrough.
Mm-hmm.
And
Mm-hmm.
we have potential breakthroughs in the lab, but they're relative breakthroughs. If it's a cool finding in my cell culture, that's less interesting than if it was in an animal model, which is less interesting than if it was in a small human study, which is less interesting than a
Right.
large clinical trial. And so, putting it in the right context, I think, is really key. And Stat News does a pretty good job of that. The other couple of sources that I really like are both Science News and Nature News. So those are the two of the premier science journals, but they have more lay public facing news articles that they put out.
Mm-hmm.
They still do a pretty good job of contextualizing the scientific discoveries for people that
Mm-hmm.
aren't experts.
So, okay, so what I hear you saying is I don't necessarily need to go and find the scientific paper and try to read the abstract
Right.
and all of that other stuff.
But go one level down.
Low, yeah.
So that
And
the context is there again.
but what if I wanted to read like a study? Like, how do you determine for a normal person like me, what is like, this is the meat of what we're trying to say?
If you did want to read a study, National Library of Congress runs PubMed, which
Mm-hmm.
is a public repository of all U.S. funded research, but it also has a lot of even non-U.S. funded research because it's a pretty prestigious repository. Articles and absolutely the abstracts are all published there. So anybody can go to PubMed and type in something that they heard, keywords, COVID, vaccine, whatever,
Mm-hmm.
and it'll just dump the whole list of all the search results. It's kind of like Google in that regard. They're not going to be filtered by the best, or there's no
Right.
reviews of the articles there, but they've all previously been peer reviewed by other scientists that were not involved in the work and then published, and the abstracts are publicly available. And so you'll typically be able to figure out some of those details, probably enough for most in the lay public, about what type of study was it. Was it, a clinical trial? What phase? Was it an animal
How
study?
many phases are there in clinical trials?
There's three phases. So there's phase 1, phase 2, and phase 3. Phase 1 is just, does it hurt people? you're, you're not asking if it works. You're just asking if it does harm. And so those are typically very small, only, 10, 20 people. And
Okay.
even those are typically, you don't just get 20 at one time and inject them. You do kind of rolling recruitment and make sure you're not killing people in the first couple. And then once you pass that and show that you don't have any major adverse events, you typically go to a phase 2, which is larger, still looking predominantly at safety, but starting to look at what we call efficacy, or basically does it work.
Mm-hmm.
but it's not really designed to definitively show that it's highly effective. And then phase 3, or what we call the pivotal trial, is really asking, does it work at scale? Like, do we see a really strong, robust effect for whatever therapeutic or vaccine that we're using? So as far as COVID vaccines go, we've got everything.
Let's talk. I was just going to say, let's talk about the
Vaccines.
Vaccines.
Yeah.
So, okay. So some of the vaccines is in the final stage.
I think right now there's just one that's at phase 3. That's phase 3 within the U.S. system, which is AstraZeneca, University of Oxford vaccine that's been in the news quite a bit.
Yeah.
They're in phase 3. There's a couple of others that are in phase 2. And then there's a Russian vaccine, and there's some Chinese vaccines as well that I'm not sure how much they're lining up with the phase 3 model.
Okay. Because you hear so much about how the FDA is allowing this to be fast tracked.
Yeah.
And when I've talked with my friends, like I was just talking with a friend about this yesterday, and she was like, well, if they get a vaccine, are you going to get it? And I was like, well, yeah. And she was like, well, what if there's something, like, what if it's not safe? You know? And I think there is a level of, like, it's questionable if the quality is going to be as good because it's not taking the normal route that a vaccine would with the FDA.
Yeah, so let's talk about that, because I think there's a lot of misunderstanding of what actually is happening. So the fast track process is a normal FDA process by itself, but right now it's actually being used as part of Operation Warp Speed, which
is that what they call it?
Yeah.
That is awesome. I didn't know that.
Yeah, so they're borrowing from, what is that? Is that Star Wars or Star Trek Warp Speed?
I think it's Star Trek, because I don't think it's Star Wars.
Okay.
The science fiction geek in me is totally excited.
There's
now.
Totally some nerd in, somewhere who's like, sneak this in, and I was like, I did my
like
job
an
today.
Easter egg.
Yeah.
Like the Disney Pixar Easter eggs.
Yeah. Yeah. The total operation is Operation Warp Speed, and they're leveraging FDA's fast track program. So the thing, I guess, just to nip right away, is that the fast track program does not lower the barrier, the safety kind of hurdles that have to be cleared for it to get to market.
It's not like cutting corners.
No, it's not cutting corners or removing hurdles from the race, if you will. Instead, it's telling you where the hurdles are and talking to you about strategies for clearing them.
Okay.
So normally,
they wouldn't share that information?
Well, so normally the way the FDA works with therapeutic companies is they do have conversations, but it's really, the FDA's job is normally not to help get a therapeutic to market. Their job is to protect the public health.
Right.
And so it's typically the company's job, who's applying for approval, to basically make the case that their therapy works and is safe, and to make a really compelling, strong argument to the FDA. And the FDA kind of sits back as a bit of a cynic and a critic and pokes holes in it, and will come back and say, we really want you to do this additional study, or we want you to do this other tox analysis, or yes, we'll approve it, but we want to be able to track this and this after you've rolled it out. And what they're doing now is basically coordinating that process so that the FDA at the get-go is saying, for these vaccines, we want your trial to look like this.
Okay.
And actually, we're going to help manage it, and we're going to fund it. And so the FDA is not standing back as a disinterested party. This is just a gatekeeper, but they're actually trying to make sure that the work done the first time is done to the standard that they expect.
Okay.
So you're getting rid of some of this kind of loops of having to redo the work, because maybe I do my initial animal study in mice, and they really want to see it done in rabbits.
Or bats.
Or bats, or whatever. probably, I don't know if they do clinical trials in bats, but, there could be things like that typically.
That was my attempt at a joke, at a COVID joke.
But, yeah, so, that's, you can imagine if I did a 6-month study and I'm in the wrong model, and the FDA then comes back and says, well, we have new evidence that now says that that's not a great model. We want you to do it in this other model. That adds a lot of time to the process.
Right.
So having them involved with the design of the trials, and not just for one vaccine, but the Operation Warp Speed, I think they started with 14 vaccine candidates, and they've narrowed it down to 7. And so they're involved in both vetting which ones they're going to get behind, as well as providing the funding to really accelerate the process along the way. So that's one, is coordination between FDA and the manufacturers, and actually designing the trials and sponsoring the trials centrally, and not having the company as a sponsor, but having Health and Human Services as a sponsor for some of these trials.
So the UK vaccine,
yeah,
do they still have to go through the same FDA process?
Anything that's marketed, sold, or manufactured in the United States has to go through the FDA.
Okay.
And so we are the largest healthcare market in the world because we have the most expensive healthcare.
Oh, yeah.
And it's one of the ones funded. So we're not just funding US-based vaccine candidates through this Warp Speed. So the Oxford vaccine with AstraZeneca, they got 1.2 billion in support from Warp Speed. Moderna's gotten 483 million, and Johnson Johnson has gotten 456 million. So we're putting tremendous amounts of money into these, not just for the clinical trial, which is expensive because in trials you're paying for the patient's entire care.
Right.
But we're also, part of this money is going to manufacturing scale up. So that's the other part of Warp Speed. Instead of, if I'm a company and I don't know if my vaccine works, I'm not going to build out my manufacturing and production until I know that the vaccine works.
Right.
In Warp Speed, they've said time's more valuable. So we're willing to pay you now to set up manufacturing and start producing before we have the phase 3 data.
Okay. And they've started the Oxford one started phase 3, hasn't
Oxford's in phase 3, yeah.
Mm-hmm.
What that'll mean is instead of finishing your phase 3 and then having to wait to figure out manufacturing and to actually produce the vaccine, and just for context, the normal seasonal influenza vaccine is about a 10-month manufacturing process to get enough doses for one season of flu vaccines.
Okay.
So
that's a long time. It's
a long time to make enough doses. Instead of having it being a linear road that, well, we're going to do this and then that, and then, show it works, and then figure out manufacturing, and then figure out scale up. They're saying, well, while we figure out if it works, let's start solving the problems in manufacturing and scale up, knowing some of it we're going to throw away. If it doesn't work, we just burned $200 million.
Gosh, I can't imagine having that kind of money.
When you're talking, yeah, I mean, but that's what, 70 cents per person in the US?
I guess so, James. I don't do math. You do. I married an accountant for a reason.
Yeah, so there's 340 million people in the United States. So yeah, it sounds like a lot of money, and it is, but
it's not really when you break it down.
Really not. It's less than a penny a day for a year. And so when you're talking about something that could potentially save, hundreds of thousands of lives over the course of the next couple of years, it's a worthwhile investment. What's different this time with Warp Speed is that normally if a company wanted to do that, it'd be the company's money.
And it's not. $200
million for a company is a lot of money to waste.
Yeah.
And what the government has said is, the government will take the risk because it's a national interest, global interest really, to have a vaccine sooner than later. So that's the big thing with Warp Speed is the money is coming in fast and hot, and everything's in parallel, but the standards aren't lowered.
Yeah, that's the key.
They're just being coached on how to do it right the first time.
Okay.
And that's really important for somebody. So Moderna is a completely novel we don't have a single vaccine like Moderna on the market right now. It's a completely novel class of vaccines. And
Okay,
so
so
there's
explain this. So
yeah,
what do
so
you
most
mean?
vaccines that we have, that you've had, that I've had, are either live vaccines, which aren't very common anymore, attenuated vaccines. So
Hmm.
live vaccine is live virus, right? It's actually giving you the virus and it'll
Isn't that
learn.
what the flu vaccine?
No,
What?
the flu is attenuated. So the flu is we grow up a bunch of influenza in chicken eggs, and then we mechanically break it up into lots of little pieces,
Okay.
and then those pieces are packaged into the vaccine and given to you. So you're given pieces of what used to be live virus, but there's no live virus left in it.
Okay.
And so that's what you'll sometimes call here an attenuated virus, is like influenza,
Hmm.
but it's made from pieces of the actual virus. There's others then that will use other viruses. So one of the things in bioengineering that's really cool is we use viruses as tools all the time. They're really simple structures, and they're really good at making copies. And they're
Hence the
easy.
reason why we get sick from them.
Yeah, yeah, they make lots of copies.
Yeah.
But we can engineer in different genetic codes into that virus backbone. And so you can take a relatively benign virus and basically force it to express the spike protein. So this is the thing that the coronavirus displays on its outside. So we
Hmm.
can take a virus that doesn't have any of the, basically the health and contagious effects of coronavirus, but just make it basically display the badge of coronavirus.
And then that
That's
gets our like immune system to like fight it.
it teaches our immune system to see the badge.
Got it.
And so when the real coronavirus comes, it says, "Hey, I recognize that. I recognize that protein," and you'll already start attacking the virus based on that. So
Okay.
those are good, and we have some vaccines already that are, I think they're adenovirus-based vaccines that do that, where they just teach your immune system to see a part of the actual virus that you're trying to fight,
Hmm.
and that's good, but it's not great. So if you can think about this, Beth, you're wearing glasses right now. If all I
Yes.
said is, she's going to be wearing glasses and that's how you'll recognize her, that's, okay. But there's many other attributes about you
Well, right.
that
So yeah. So how does that work then
yeah. So
with the vaccine?
those vaccines are, if you happen to take your glasses off or change your glasses, that'd be a mutation. And then the
Hmm.
vaccine's not going to work as well. The other bit that we worry about from a vaccination perspective is your immune system normally learns multiple aspects of the virus, not just one. So just like I see you, not just as a person with glasses, but all your other features,
Hmm.
ideally you want your immune system to learn all those features.
Right.
And so those are basically trade-offs that you make when you do just, well, I'm going to select just the spike protein. Now, it's not a blind process. Typically what scientists do is they look at, well, what piece of the virus does your body normally recognize? And they also will look at what parts of the virus mutate the slowest. And so they'll look
Wow.
for the overlap of that, of what is your immune system good at recognizing and what doesn't mutate very often. And they'll
Hmm.
pick that as the thing to present to your body in the vaccine
Hmm.
for the best
And
chance
so
of
is
getting a
that what
durable.
Moderna is doing?
So Moderna is doing something very different. And so
So that's the normal
those
way.
are all normal. So live
Okay.
virus, not very common. Chopped up virus, like think about putting a bunch of virus in a blender, a really fancy blender, and just obliterating it to tiny little fragments.
Yes.
That's like your
Not a
influenza.
okay. Okay.
Then the third is asking a benign virus to make a piece of the nasty virus so that your body
Uh-huh.
can learn to see it. And then what Moderna is doing, instead of having a virus make that thing,
Yeah.
they're actually telling your own cells to do it. So it's an mRNA-based virus. So just a recap of high school biology.
Which I
Um,
don't remember at all. So I
so
guess it's a good thing.
we have what we call the central dogma of biology, which is we all have DNA in our cells.
Yeah.
And we say, well, that encodes everything. Well, it encodes it, but it goes through first the process of being transcribed into RNA first. So we go from DNA to RNA. And then RNA are the actual transcripts that then get turned into, or really they're the instructions for proteins.
Okay.
So DNA to RNA to protein is the central dogma.
And the protein is what again? This is
Protein
all
is,
so
yeah,
stinking confusing, James.
the protein is what most things are actually made of. So
Got it.
DNA and RNA are just instructions.
Okay.
Protein is literally the meat on your bones is all protein. I mean,
So
your
it's like
skin is
looking
protein.
at an piece of IKEA furniture.
Yeah, but it's like if there was a catalog there's somewhere a catalog in Norwegian of all
Yeah.
all IKEA furniture, and it first needs to be transcribed into English onto a single piece of paper.
Uh-huh.
That would be the RNA.
Okay.
And then you build the furniture based off of that. So your
Got it.
furniture would be the protein.
Okay.
And so what Moderna is doing that's different is instead of having a virus basically force your cells to make that, they're actually giving your cells a copy of that RNA so that you yourself will make the spike protein. Your own cells will make the spike protein.
But okay, so because with the coronavirus, the reason why it's called corona is because of the spike, the crown, right? That the cell has.
Coronaviruses have yes.
Yeah, they have a crown.
Yep.
So is the crown itself bad, or just what's encoded in the crown that makes it dangerous for us?
Yeah, so this is complicated because it's a little bit of both.
So let's pretend that you're talking to your 5-year-old daughter.
She's really smart.
She is really smart. Let's pretend you're talking to a 5-year-old me who does not care about this stuff, but really does care about it because it matters now.
Yeah, so the spike protein is part of what latches on to your cells and allows the virus to actually invade.
Got it.
And so
because it's like a sword.
not exactly. It's not a physical attack. It's more of a counterfeit key.
It's not as dramatic as I'm making it.
No, it binds something else called the ACE2 receptors in your cells. I know you don't want to talk about.
Nope, I don't.
So it's not slashing its way into your cells, but it's basically using a back door to get into your cells.
So it's sneaky.
Yeah, all viruses are sneaky.
They are sneaky.
And then there's really two aspects with coronavirus. There's what the virus does itself, but then there's your body's response to the virus. And a lot of the really severe cases we're seeing is the level of the immune response against the virus is overwhelming and is starting to do a lot of damage itself. So there's cytokine storm, which is basically just, how terrible you feel if you have actual influenza.
Yeah.
Like days of fever and you just feel like death.
Mm-hmm.
You've got a lot of, basically, your immune cells are just screaming.
The whole time.
And they scream in the form of basically producing chemicals called cytokines. They make you feel really terrible.
Okay.
But they also recruit an even larger inflammatory response. And there's normally, checks in your body that will start to, resolve that inflammatory response. But if it gets out of control is where you end up in really a bad situation.
Like on a respirator.
Yeah. Yeah.
So it's kind of like your immune system is going overboard because it has no idea how to get things back to normal.
Or there's so much damage happening that you just keep recruiting more and more of an immune response. And if you think about, even think about a normal, like let's just take like a bee sting, right? Or bad like mosquito bite.
Yeah.
What happens to where you get bit? Like it gets really red. And so that's a bunch of blood and immune cells that just came. Mm-hmm. It swells. So you've got fluid coming into the system. Mm-hmm. And then you feel all the itching and the pain. That's all cytokines and other chemicals that are been produced to say, hey, like, this isn't right. Like I need help fighting whatever is happening. Now imagine that you have all those bee stings on your lung.
Oh, yeah.
And you have those same things happening. If you have inflammatory infiltration, you've got a bunch of fluid coming in. That's a really bad one.
Right.
And you have then further chemical signals that are recruiting even more of a response.
Uh-huh.
And so that's where people get in a really bad situation. When your lung tissue gets badly inflamed, then it can't really it's not functioning as a lung anymore, because there's all this fluid and, pus and mucus and stuff in the way that's not letting gas get to where it needs to.
Mm-hmm. Mm-hmm. That makes a lot of sense.
So Moderna is brand new.
Right.
So the need to really coordinate with the FDA is really necessary, because
Yeah.
we've never been down the road of this type of a vaccine before. What's cool about it is that it's way easier to manufacture, because since it's an RNA, we can make that without any biological system. We don't need cells to make it. We don't need chicken eggs to make it.
Okay.
Synthesizing DNA, RNA just with chemistry is
Mm-hmm.
that science is down. And so
Yeah.
you can crank out product really fast without actually having to rely on a bunch of cell culture or a bunch, for the flu vaccine, we use millions and millions and millions of chicken eggs. Like we have a government stockpile of chicken eggs. The location is secret.
No way.
In case we have a new, highly, virulent, lethal influenza, yeah, we have a government stockpile of chicken eggs that is constantly replenished as they expire.
Wow.
And it is a national security secret of its location.
How would you like to be in charge of that? The national stockpile
just
of
like
chicken eggs.
a warehouse on a mountain somewhere filled with eggs.
Right. Yeah. And you can't tell anybody what you do.
Yeah. Just going to work. Yeah. So having the ability to just make it with machines provides both flexibility and scale that we currently don't. I mean, because right now, if an actual avian flu, Iowa's the leading egg producer in the country. And so you'll hear about this occasionally, that there's a really nasty virus that goes through the flock, and, suddenly there's only, a half as many or a third as many chickens as there were before, because there's just a massive disease went through. And so that has direct public health and national security implications if you're reliant on eggs for your public health.
Okay, so
with Moderna, if you can do it with machines, you're no longer at risk of just losing everything by somebody wiping out your chicken flock.
Right. Okay, that makes a lot of sense. So now with the vaccine, so they're in the last phase. Oxford
last phase. Others are marching on, starting phase 3. It's supposed to be, I think, like late July.
And so are they actually infecting? How do you test if it's if it
let's
works?
Talk about the types of clinical trials.
Yes, let's, because I want to know what crazy people, in my mind, I'm thinking, what crazy people are trying to get COVID?
Yeah. So what you're thinking of in your mind, we call a challenge trial.
Oh.
Which is where you give some people a vaccine and some people a placebo, and then you challenge them with live virus. I'm not aware of a single COVID vaccine that's being challenged. So in part because, and just to get it out there, the history of ethics in medicine is bad.
Okay.
The history is really bad. And so, the silver lining in that is that everybody, even my 19-year-old second-year students, go through ethics training, learning about all of these literal crimes against humanity that have been done in the past, and then what actual ethics is and our burden to protect the public, and so this would be considered, to knowingly infect somebody with a potentially deadly virus is highly unethical.
Yeah, I would say so.
We don't
fingers crossed that you don't actually die. Nobody would sign up for that.
So you don't do challenge trials for lethal diseases. I mean, if it was literally a common cold, sure, do a challenge trial. The nice thing with a challenge is that they're small, because you know exactly who's been exposed.
Okay.
Instead, what they're doing is they're recruiting people to be in either the actual group or placebo. And again, you don't know which one you're in.
Right.
But the recruitment is strategic. So the way these trials run is that they'll run until a certain number of people in one of the groups test positive, and then they'll stop.
Okay.
So to ever finish your trial, you need to have it run in a place where there's an active outbreak.
Okay.
So AstraZeneca, I think they have sites right now somewhere in the UK. I know they have a South Africa arm, but they have basically arms of the study that are happening around the world, but they're all focused in places where there's a current active outbreak.
Okay.
So they're more likely to actually hit finally that endpoint of we have 500 people in this group that got infected, and now we can ask, well, how many were in the other group that were infected, and which one was better, and how much was the difference, and all that. So those are challenging with a virus like this, because as we've seen, it's been moving, right? So 2 months ago, you would have planned your trial for New York City.
Right.
You wouldn't do that right now.
No.
Because the measures in New York City have been really effective at bringing it under control. But now you'd go to Florida.
Right.
And there
or
are
Arizona.
Arizona, or Texas, Houston area would be a really great one right now. Like they're getting hammered. And so you'd have a really good shot of actually being able to tell, does this vaccine make a difference amidst an outbreak?
Got it.
And just from a historical note, for those of us that are in our 30s, you might remember SARS.
Yes.
So SARS was 2003, 2004, another coronavirus outbreak. We developed a SARS vaccine, but by the time it was developed, there was no outbreak in the world to test it on.
Which I don't like. So how long was SARS around? Like, was it considered dangerous? Because I don't remember it. Did it ever get to the U.S.? Because I don't remember.
It never got to the Americas. No,
right.
It was a much more lethal and more symptomatic disease in that you could, and it was bad in that, for those that got it, it killed a lot more. I think it was about a 10% case fatality rate compared to we're seeing like 1.5 to 2% for COVID. But the big difference is when you had SARS, you were really sick. Like everybody was really sick.
I remember you saying this, like you could not move and walk around.
Yeah, same with Ebola. Like Ebola was really lethal, but like there's no silent carrier of Ebola. Like you're terribly sick. And so
it's the silent, the asymptomatic people.
That's the sweet spot of this virus that we talked about last time, of it's that you can walk around with it, or even maybe you do eventually die of it, but for 2 weeks you can walk around spewing viral particles and not know it until you finally are symptomatic. So anyway, we spent all this money on a SARS vaccine and developed a good candidate that seemed to work, and then didn't have people to test it in.
Okay.
And that wasn't all wasted, because that was really the starting point for knowing that the spike protein is a really good thing to target and all that, that led into the current developments with this virus. That's why the vaccine locations are always going to choose places with viruses currently really running rampant.
Got it.
That's all we have. Thank you so much for listening. Be sure to connect with me on bertmanderson.com, and please visit our website, matriarchdm.com, for all of the show notes and to hear other awesome podcasts.